An ontology is a tree. It's a tree which represents in a conceptual form the organisation of whatever you're talking about. You can have language trees, language ontologies, and you can have ontologies of General Motors cars. You know, they all have their branches and so on. One of the issues in doing this is that is how complicated do you want the tree to be? And when we started off, Lewis was actually happy with the idea of having 6 levels, starting from the brain vesicles. We could go through 6 levels of subdivision, and the leaves, if you turn it upside down, which are now at the top, would be the individual brain structures, like the hypoglossal nucleus or the red nucleus or something of that kind. And it's that 6-level tree which he developed for the chick, and which we've applied to the rat and mouse, and now to the rhesus monkey. But Lewis wasn't to be stopped there, and his work has continued, and the Allen Institute hired Lewis a couple of years ago to map their developing mouse brain atlases, which, if you haven't seen that on the Allen site, are just a superb collection of sagittal atlases of the developing mouse brain with the entire ontology in place. And so really you could say that the only place that you can find the mammalian brain ontology at the moment besides BrainNavigator is the Allen site. But at that site, he's only taken it down to the first 5 of what are now 13 levels. Why 13 levels? Because if you're a passionate embryologist, as Lewis is, you want to, in fact, demonstrate a branching at every significant division point in the organisation, so that when you get dorsal ventral split, or when you get ventricular peel splits for every structure, he wants to divide them up in that basis. I don't think it's saleable to the general populace to be able to divide everything through that 13-level split for daily usage and things of that kind. And I think probably Lewis would agree too that we'll have to have this compromised, compressed 6-level version in order for kind of daily use. But if you want to see the entire story, the 13-level organisation does give the entire story better than the 6th level does. So Lewis has prepared this series of illustrations to show the way that you progressively move down at least to layer 5, and then I'll tell you about the other ones, which is on the Allen brain site, and these are the pictures. So that level 00 is the unpatterned neural plate at the top left of the top right of the picture there, as you see it, and then at level 1, the proto segments are split up, so you can see the difference between the spinal cord, the hindbrain, midbrain, and forebrain. In a conversation with Doug Bowden just then, we were talking about the fact that when people talk about dividing up the parts of the brain, very often they leave off the spinal cord and the retina because people think the brain is the brain. You just chop off those 2 bits and you have the bit in the middle. And it is important that we make sure that the spinal cord ontology considerations are taken into account in this as well. And actually they turn out to be quite interesting. As I'll try and mention just towards the end, but also that you have to have the outgrowth from the floor plate going out towards the eye, so you need the retina as well. But as you can see here, at level 2, you get some of the secondary divisions in the forebrain and hindbrain. Now, Lewis has in this particular arrangement split the hindbrain up into 4 regions, which later demonstrate the rhombomeric subdivisions. He's attempted to do this not because you can see boundaries between those 4 regions, but to have some clustering which might help nomenclature in the future, because the existence of 11 rhombomeres in the isthmus doing the whole of the hindbrain creates some difficulties for people who are used to pons and medulla and simple brainstem nomenclature, and there's going to have to be some clustering. I'm not exactly sure what the clusters ought to be, but this at least can be overlapped with the pons medulla story that we take from the human brain. So that Level 3 is where it starts to become really interesting because the neuromeric or segmental subdivisions can be seen here. And I suppose just to make a couple of comments on these, the first segmentation system which was appreciated was that in the hindbrain. And going back to the late 80s, early 90s, was demonstrated in the chick that there were clearly separate subdivisions which were called rhombomeres, because that was the term that Hiss coined 100 years before, the segments of the hindbrain, and 7 of those were originally defined, and because there are no embryonic bumps for the last 4 of them, Krumlauf and others have dismissed the idea that there might be any more. It is strange that the end of the hindbrain is exactly 4 segments long, and there are postotic segments there which lie alongside it, which seem that it's a fair clue that there might be. But Lewis has demonstrated with grafting experiments in the chick, and now with Hox in situ labelling, that there are in fact not 7 but 11 rhombomeres. He used to call the last ones pseudo-rhombomeres, but they're rhombomeres, just that they don't have very early embryonic bumps, such as Hiss described in the rostral part. And the other important thing is that between rhombomere 1 and the midbrain, you can see that M1 on level 3, there's another area squeezed in there, which is the isthmus. And this is a bit of a problem for most mammalian neuroanatomists, because you look up the majority of neuroanatomy textbooks, in fact I'd say 98%, and the word isthmus doesn't appear if they're mammalian neuroanatomy textbooks. And the reason is that people think that it's either part of the midbrain or in fact it's part of R1, and many researchers talk about greater R1. It's interesting, it's just twice the length of another rhombomere, which might have been a clue, but nonetheless people have ignored that. And but in birds and reptiles and fish, everyone knows the isthmus. They're quite a big chunk. You know, it sits between the midbrain and the hindbrain. In fact, it's the location of the very important isthmic organizer for those of you that are involved in development, where FGF8 is expressed. I can tell you that we've now got a lovely FGF8 Cree lineage in mammals, which shows you the exact position of the isthmic strip, and it's just where you would anticipate it was on the basis of homology with a chick. The other thing that's happened is that in the diencephalon, in the region here where we used to have diencephalon just there, we've now got P1 and P2 and P3. This subdivision system was developed by John Rubinstein and Louis Puelles. They originally tried to have 6 of these prosomeres. They eventually found out that it was really a discontinuity between the diencephalon and the remaining parts of the forebrain, and so they retracted back to just 3, leaving the rest of it to the remaining parts of the telencephalon. And these 3 parts, which we normally would recognise as pretectum, thalamus, principally thalamus, and prethalamus, sit really caudal to the rest of the forebrain, and as it develops, you'll see that with that bend that I described before, is that these 3 prosomeres sit dorsal to the hypothalamus, which is more rostral to that in the forebrain. People have looked to try and divide the midbrain up to see if it had segments too, and you'll see a very small caudal segment, which is often called the preisthmic part of the midbrain, but largely it's defied division into further segmental parts and stands as a pretty much unitary structure. One thing I will just refer to at this stage is that people who know about the spinal cord will know that when you look at books about the spinal cord, that the spinal cord is supposed to have, depending on the particular species, anything from 32 to 34 segments. The interesting anomaly here is that you don't see 34 segments in development. And in fact, there are never 34 segments. The only reason that the spinal cord has 34 segments is that there are that many vertebrae and only so many spaces for the nerves to get out. And so segmentation is imposed on the spinal cord. Now, I will tell you later on, it does actually have some segments which are genetically defined, but there are only between 6 and 8 such segments down the spinal cord. So the new spinal cord ontology will have segments, but not the ones we previously thought. They're large strips, which are regional strips defined by the HOX genes, whether it's HOX6 or HOX9 or HOX5, so on through that. So to move to the next 2 layers, which are where all the major parts of the brain are essentially set up, shows you we've already seen level 3, and at level 4, the alar basal differentiation splits all of these areas up. And that's fairly easy to see in the hindbrain and indeed in the midbrain, where the alar basal split is convincing. But of course, as you come round this corner here and drop down into the hypothalamus, the alar basal split is a rostrocaudal split. And that's what's confused people in the past, and no wonder. I mean, it just is not intuitive in the way that should operate. And so that we have different parts, the peduncular and prepeduncular parts of the hypothalamus, which are basal plate, and we have what is essentially an alar plate region just in front of that, and then we have the further development of the telencephalon. As far as the I think one quite interesting thing about this is that because we have an alar basal split in the in P1, P2, and P3, that's pretectum, thalamus, and prethalamus, that these 3 parts must make it to the C. You know, they've got to get right down to the ventral peel surface, and people often forget that when they're trying to draw these structures embryologically, that you can't jump from the hypothalamus to the midbrain in one step because you've got the 3 parts of the diencephalon in between. And one of the oddities of this is that the substantia nigra, which curls right around this split there, from isthmus around to P3, fills in part of that basal region in all of those parts. It's not just in the midbrain, goes back to the isthmus, comes right forward to P3. And lastly, we get to the last picture, which appears on the Allen site, which is Lewis's version of how the whole brain is starting to be set up before the final processes which are concerned. And you'll see now we have pretty much of a complicated matrix, because down in the hindbrain we don't only have alar and basal, we also have floor plate and roof plate areas to consider. And actually between alar and basal there's actually a thin line too, which is referred to as the liminal region. So you actually have 5 layers through that. And when you start to classify structures down in the hindbrain, you have to allocate them developmentally to one or other of those.